I wanted to start by telling you a story. I was on a plane maybe 6 years ago, and I was sitting beside someone who was a financial advisor. And he was telling me lots of great stories about things he had done and money he had made for people, lives he had enriched. He was a bright guy, and he had done a lot of really clever things. And I was beginning to think, he's really smart. He is making people's lives better. And he said to me at some point, he got tired of talking about himself, and he looked at me and he said, well, what do you do? And I said, well, I'm a plant pathologist. And he said, do you think plants are important? And I said, well, I exaggerated a little bit, and I said they are the basis of all life on Earth. There are some organisms in the deep sea that are not really plants, but nonetheless, plants are the basis of all life on Earth. And this man did not know that. And so I had to explain it to him, and he was really quite skeptical. But it's for this reason that I joined Penn State 18 months ago, because I felt like I needed to help train plant doctors. And that's what we do in our department. We are the plant doctors. And if you don't know it, plants get sick too. In fact, they get viruses, just like the cold virus you get. They get fungi, just like your athlete's foot. They get them too. They get worms. Anybody want to eat these carrots? Kind of wormy carrots. And mistletoe, something that we really love around Christmas time and makes us heartfelt and warm and cuddly with one another. It's a parasitic plant that attacks plants. Well, I study bacterial diseases of plants, and there's all different kinds. And in fact, these are some galls that are really important to business all over the world now. That restrict water flow going up through plants. These wilts that you see here in tomatoes that are important, causing diseases all over the world as well. We have spots on our apples and our vegetables and our mushrooms caused by bacteria. Also rots. Ever have stinking potatoes, rotten potatoes in your storage cabinet? That's something I did my PhD on, in fact. These rots, in particular, this banana rot and potato rot, you know, these are staples of people all over the world. And, you know, David did a great job of teeing me up to talk about plant diseases, because these really are the things that are making a difference in people's lives. And fields of crops, this is a field in California. I don't know if you can see this, but these brown patches throughout, this is a type of broccoli or crucifer that's grown in California and that we eat in our family all the time. This particular field, the farmer had to till under because of all of the disease. So that fossil fuel we just heard about, it went out to put the field out. You know, they planted the seed, they fertilized it. All of that input they had to till under because of the plant disease. We don't want that wasted energy. So plant pathologists are working around the clock to really try to solve some of these problems. So one of the things that is remarkable to me is they do that. They kill lots of plants and they damage these organisms, but they're tiny. So this is the top of a pin that has been magnified. So if you think about that pin and then all of those little orange bacteria there, it's kind of hard to see them. So it's really hard for me to work with things I can't see. Well, luckily, I have a couple of videos here. Whoops. Where'd it go? Luckily, we can watch them as they divide on a petri dish. Okay, so this is bacteria dividing, and what happens with bacteria is they absorb the nutrients, they get bigger, and when they get big enough, they divide in two. And I wish I could do that because I'd eat more ice cream, right? These are bacteria then on the petri dish, and that's what's happened with these bacteria. A single bacterium has landed on that nutrient source, that food, and it began to divide, and it made a big pile of bacteria, which we can now see. And that's really the basis of a lot of the work that we do with bacteria, is growing them on petri dishes and then watching them, studying them after that. Well, they also grow and divide on plants. And this is the picture, this is a diagram of what a plant leaf might look like. And bacteria grow on the surface and pick up the nutrients from the leaf, but they also can colonize the inside of the natural openings of the plant, or if, let's say, hail damages a plant, that releases some of those nutrients as well for them to colonize. They then can cause the disease in some cases. And then a farmer, she'll have to till that field under, as I talked about before, because can't sell it, right? Well, what's interesting is this tissue protects the bacteria. If the bacteria from the leaf tissue is left to the soil on its own, it'll be degraded. But if it's protected by the leaf tissue, it won't. So as the leaf breaks down and releases the bacteria into the soil, it's then attacked by other microorganisms. So the bacteria attack the plants, but there is something that is attacking the bacteria too. And one of the things we study that attack bacteria are viruses. These are bacteriophage. They will inject their DNA into the bacteria, hijack their ability to make proteins and nucleic acids, the substance of what the cells are, and it will cause the cell to make more of itself. And then it'll burst the cell open and release itself into the environment. That's what's happening here. I don't know if you can see it. This is a lawn of bacteria, and these little circular holes there are where the bacteria have all been killed by the virus. Okay, and I'm all about whodunit, I have to say. So this is another picture of that, and we've actually tricked the virus to produce light when it enters. And that way, by measuring the light, we can tell where the bacteria are even on the plant. And here's an example of just seeing these circles here and then putting it under a dark field, and you can see the light from those bacteria. And so this is one of our detection mechanisms, so we can see where they are before they do it. Another group of, I want to tell you about where we're using this. So we're studying mushroom diseases. This is bacterial blotch in Pennsylvania. There are approximately 8 different species that cause this, 6 different species that cause this disease. And in fact, we don't know which ones are causing the disease in any given field. People in the past have used bacteriophage, but they've only used bacteriophage to one species. And so what we're doing is we're looking at a cocktail of bacteria, because if we only attack the bacteria that are on that mushroom, then the bacteria that are on this mushroom will explode because the others are under control. And if we control that one, it's kind of like mushroom whack-a-mole, then the bacteria on that particular mushroom will explode. And so if we can make a cocktail of these and understand who is causing the disease in this system, we can really strategize which cocktail to use in this particular system. So another group of organisms that we work with are myxobacteria, and these myxobacteria are called the wolf pack, or the hunters of the bacteria. So this is E. coli, a colony of E. coli here, and these are the myxobacteria. They go out, and they go out as individuals to try to find other bacteria. And once they find them, they come in and surround the bacteria, and they hunt like a wolf pack. And so what they're doing here is they're moving into that E. coli colony, they're producing enzymes that lyse those organisms open, and then they slurp up everything that was inside those other organisms and produce more of themselves. And these are different kinds of myxobacteria. They almost look like fungi. They're so cool. And we really enjoy working with these. So what's the point of all of this is that we actually can use these organisms to control disease. So this is a water control. There's absolutely no pathogen in there. Here we have the pathogen and nothing done to these lettuce plants. And you can see the pathogen kills the plants. And then in these plants, we've treated them with myxobacteria. And in fact, they do control the disease. And so taking this to the field would be a good idea. But what we're really interested in is understanding how these organisms interact in the environment without us adding it. And how can we increase the populations of some of these that will be beneficial in controlling the organisms that we're really interested in. Even food pathogens of humans that might be out there, for example. So currently, as I said before, what we do is we isolate the bacteria and we name them and we say who they are and we say where they are. And that's kind of like this picture. If we were at a park where people would be swimming and we were trying to describe what they were doing, and we said, okay, we've got 20 blankets, we've got 20 umbrellas, we've got 30 people. This is the kind of study we initially were doing. But through the power of sequencing and being able to detect molecules in the environment, both proteins and other kinds of molecules, we're now beginning to really get a clear picture of what the community of organisms would look like, even though we can't see them. So through these techniques, we're able to describe how the organisms are interacting, rather than just who's there. And so that's what we're doing at the Microbiome Center. So my department and a number of departments and colleges around the campus have been involved in pulling together all of the scientists who are artists, as well as people working in mechanics, mechanical engineering, for example, pulling them together to have discussions about how we can study microorganisms, their environment, and other organisms, and the system rather than just the individuals. And that's what we're doing today here at Penn State. So thank you for your time today. I'll just say one other thing. If you've got a plant disease problem, we have a plant disease clinic. See me afterwards, I'll give you a card.